that I get to invite up one of our superstar presenters, Professor Ezio Bonifacio, who's the director of the Center for Regenerative Therapies at the University in Dresden. And he's really the international authority on the autoantibodies that serve as predictors for type 1 diabetes onset. And started his early independent career at the Raphael Scientific Institute in Milan, where I first met him. It's really decades worth of study and work. Ezio currently serves as director of the Center for Regenerative Therapies in Dresden, and he has played a central role in understanding autoantibodies as a predictor of type 1 diabetes. With that, it's my pleasure to introduce my friend and colleague, Professor Ezio Bonifacio.
I thank very much both Dave and Dale and Mike, the organizing committee, for inviting me. I was also fortunate enough to spend many years with an Italian who discovered islet cell autoantibodies, and that was Franco Bertazzo, and he discovered them in 1974. I sort of took that over from him, and I think I have to thank him for many things. One is because he did bring me to type 1 diabetes, along with the JDRF, who are also here. And Franco was an entertainer, and he would introduce himself or the story of ICA sometimes in this manner. He would say, you know, who can tell me what that is? Of course, if you fly into Venice, and he came from Venice, if you fly into Venice at night with green-tinted sunglasses, this is what it looks like. And then he would show that, and that's what islet cell antibodies look like under a microscope. And he would always say, see, only a Venetian could discover islet cell antibodies. And that's really how I started in this, looking down the microscope. The important discoveries thereafter were that, first of all, they did precede type 1 diabetes, and second, that they, of course, were antibodies that targeted special proteins, and these proteins were sitting in the beta cell. There were a number of proteins that sit in the secretory granule, so this is a very important part of this beta cell, and these are insulin and proinsulin, so the key hormones, a number of proteins in the membrane, IA2, IA2 beta, which is related to that, and zinc transporter 8, and then this other molecule recently found, tetraspanin 7, and then a very important molecule called GAD65, which is also in certain brain cells as well. So these were our key targets that helped us understand a process that is happening very early in life. The antibodies don't actually cause the disease, okay, but they're superb markers of the disease or something that's going on. These are the main ones on the left, but many of you will know that type 1 diabetes is an autoimmune disease, and it is associated with a number of other autoimmune diseases. And they include thyroid autoimmunity. Often going together with thyroid autoimmunity is an autoimmune disease called vitiligo. There's also intestinal autoimmunity that's sometimes associated with that, in the form of celiac disease, and they have antibodies to a protein called transglutaminase. And then also associated with type 1 diabetes, thyroid disease, etc., is antibodies to the stomach in terms of gastroparietal cell autoimmunity. So it's sort of a complex that can appear sometimes in one form, sometimes in another form. And part of the reasons that these, or perhaps the main reason these diseases tend to associate with each other, is because they share certain genetic susceptibility. So this is one of these colourful but perhaps complicated slides that you'll see many times about the genetics of a disease, and this is genetics of type 1 diabetes. And so the higher this bar, the more that particular gene down there influences the likelihood that someone will get type 1 diabetes. And there's very many genes that do that. There's one complex in particular which is called the HLA region and HLA genes, and here you can already see that there is some similarities between some of those diseases I talked about. So if you happen to have something called DR44, and we're all very different from each other in these, but if you have DR44, you're very likely to get, well, high risk to get type 1 diabetes. A DR34 is type 1 diabetes, but you also have a risk for thyroid autoimmunity and celiac disease. A DR33 is very much celiac disease is your risk, and you may also develop thyroid autoimmunity and type 1 diabetes. So HLA is very, very determinant and also can distinguish which side of which disease you're likely to get. There's a whole bunch of what we call non-HLA genes, and you can see them all here. And some of these, for example, insulin, the gene itself affects the likelihood of getting the disease, and in this particular case, insulin really only affects type 1 diabetes, the gene. And then there's a whole series of other ones that determine the immune response or a response to infection, etc. And these are, of course, determinant in many of these other diseases. So this is one of the reasons why, or the main reason why these diseases are associated with each other. How do they increase the risk? Well, two areas that are considered. One is because they work through an organ called the thymus, and there are important people here working on the thymus, and the thymus is a place where the immune system learns what to do. And many cells, the lymphocytes that do the job, go through the thymus, and most of them are actually stop there and don't get through. And we have these cells that can recognize our own proteins in our own tissues, and these ones should not really get through, or if they get through, they shouldn't really be doing much damage. And what happens with certain genetic susceptibility, and HLA and the insulin gene are examples of this, they actually determine how many of these autoreactive cells come out of the thymus. So if you've got the high risk gene code, you will put more out. If you have a low risk one, you'll put very few out. So that's one way that the genetics works, and the other way is the immune response per se. And in general, for autoimmunity, it is such that the genetic code that you have for all these genes sort of increases the response and decreases the control of responses. So that's how these genes work. And then we know that there's an important component of environment, and the environment per se actually affects, we think, that it affects, modulates how the genes work here and here. So I'm going to talk about the autoantibodies, and much of the information that I have gained through autoantibodies is coming from these so-called prospective from birth studies. So the first one is the baby diet study. This is a German study, and Annette, who will be speaking later, was the founder of these studies. So this is one of her major contributions, and this is really following children from birth, started in 1989. Children who have a mother, father, or sibling with type 1 diabetes, they have an increased risk for type 1 diabetes. And these have, what's very nice about this, there's no other genetic selection apart from the first family history. And now the oldest child that's followed is 29 years old, which is very impressive. And the second of these is the TEDdy study, which is, I think Annette calls it the second generation of these studies. And this is, many of you will have heard about it. It's an NIH study. It involves cohorts from the USA and Europe, and it selected children with a HLA risk, so they have a genetic risk, and it has very intense follow-up. Every 3 months, blood samples, every month, stool samples, etc. And the oldest of these will be 15 in September. And there are other studies that were also followed and modelled on the baby diet, which are the Finnish study in DIP and the study in Colorado, DAISY, that we also collaborate with. So this is a wonderful example of a child who developed type 1 diabetes from the TEDdy study, where there's antibody measurement at multiple time points shown here. These are the antibodies to insulin, GAD, IA2, zinc transporter 8 running there. The ones in red are so-called positive, above threshold, and the yellow is where they first come up. So this particular child, we say, started disease somewhere before age 2, where we see insulin autoantibodies coming up. A few weeks later, the child had developed other autoantibodies, GAD, IA2 antibodies. Some months later, also zinc transporter 8 antibodies. We see, in fact, in this particular child, the insulin autoantibodies go away, the other ones persist. And as you can see by the age, it takes a couple of years. And eventually this child had an impaired fasting glucose. If you know about the HbA1c, this is not quite normal, 5.7%. Eventually also impaired glucose tolerance, which is a very difficult thing to measure. As you can see, it's fluctuating up and down here. The HbA1c still slightly going up. And eventually at age 6, developed clinical type 1 diabetes. So this is a wonderful example. It shows this first appearance of insulin autoantibodies that can go away. The stage of having many of these antibodies rarely goes away. We rarely see that change. And with this long phase of years before there's eventually a metabolic detection of a pre-disease and eventually clinical disease. The timing of that child is also pretty typical. This is what we found in the baby diab study here on the left-hand side, where we talk about the incidence of new cases that develop this autoimmunity. So these children are followed at regular time points, and then we see how many of those develop it for the first time. And you can see that in this particular study, in the German study, there was a peak somewhere around 1 to 2 years of age. So these are new cases of autoimmunity, and then less new cases as it gets older. So this is fascinating. It says that there's something happening earlier than this that's causing this autoimmunity to start. The TEDdy study shows something very similar. The TEDdy study is beautiful because it has 3 monthly samples, so you can get a very fine measure of when this is occurring, but you see this peak again around about 1 year of age. The TEDdy study has also done this in a manner that looks at which of the autoantibodies comes first, and as I showed you in that example, insulin autoantibodies is one and very common one that happens first. And this first peak is exactly that. These children develop antibodies to insulin first, the first one we see, and then other ones. There are also children who develop antibodies to GAD, which is the other big antigen. And they happen to be a little bit later and flatter throughout. So, and they have slightly different genetics associated with them. So, you know, it could also be 2 forms of the disease in terms of what causes it and how it starts. Back to the other antibodies, and I find this data in this slide really fascinating. So this is the islet antibodies, the diabetes ones. These are the ones that are associated with celiac disease. They start to happen about a year later as compared to the diabetes ones. These are the same children in the German study, the same children, same 2,000 plus children that have been followed. And this is thyroid. So here we have thyroid autoimmunity in the same children, and this is now the incidence, and they're happening somewhere late in puberty and in teenage years. So this is very nice for me because it says, okay, well, celiac disease is something we know a lot about. We know that gluten is really important in that. Okay, but you introduce gluten somewhere back here, and then we don't see the antibodies until a year or so later. So there's got to be something else that's in there. It's not just because the child sees gluten and then immediately gets disease. So there's got to be something else that's causing that. The other nice thing is that thyroid, so these, again, they have genetic susceptibility for all of these, but the thyroid autoimmunity is happening somewhere here, and we know that this is an important period for the thyroid to start, doing different types of activity. So we think that for the diabetes, this period is important and tells us that it's something that's happening right at that beta cell, that it's not just the immune system that's having a hard time, it's also the beta cell that's contributing to this disease. Well, before I go to that slide, you're going to ask me, all right, is it the same people that get all of these? And in fact, there is some overlap. There is some overlap. So this is a Venn diagram showing those who have islet antibodies, those who have transglutaminase, those who have thyroid peroxidase. But the overlap is not terribly large. So it's different people who are getting these antibodies. And that means that overall, if you happen to be born into a family with type 1 diabetes, your risk for autoimmunity is quite high. How high is that? So this is a so-called life table. This is where every slight rise there is a new child that's developing, in this case, islet autoantibodies. And so you can see by age 20, 25 years, it's somewhere around 8% of these children who have a mother, father, brother or sister with type 1 diabetes. 8% of them have developed islet autoantibodies. If you then take the thyroid, transglutaminase, which are the celiac ones, and islet together, you see that the curve looks like that. So this is 25% now. That's 1 in 4 of these children, by the time that they're 25, will have some sort of autoimmunity. That's quite impressive. If you then take those 3 HLA that I talked about, DR344433, it's half. So half of them will develop some form of autoimmunity. And if you remember that slide that I showed you before, the age at which they develop is, of course, different. So in the first 5 years of life, you're likely, if you are these HLAs, to develop islet antibodies or celiac-associated antibodies. If you get to be negative at age 10, you now have another 10, 15% risk to develop thyroid autoimmunity in your life. Age is also important because as you get older, I just mentioned it before, as a child gets older, the risk for a certain disease changes. So this is so-called landmark modeling of risk for type 1 diabetes, and this is for autoantibodies, something we've done recently on the German cohort. So this is the development of islet autoantibodies over time if you're from birth. And you can see this red line goes up, and we have a certain risk, almost 8% by age 20. If you happen to still be negative at age 3 years, your risk is now that, and you've reduced it by about half, etc., etc. So the longer you go, and if you get over that, especially if you get over that first peak, your risk is now much reduced, which is nice. It's very nice to be able to tell a family, hey, get to 4 years, your risk is half. That's what we need to do for you. And you can, it's interesting if you plot these risks over age, this curve actually fits something that's called an exponential decay, which is sort of like you get hit and then something decays. So we can discuss and think about what that might mean in terms of what might cause the disease later if you're interested. So this is, I think, really important, and what it says is if we can actually prevent the autoantibodies in this first 3 years of life, we've halved the risk. That would be pretty good. All right, if you develop the autoantibodies, will you get diabetes? And the answer is if you develop multiple antibodies, so insulin, GAD, IO2, zinc transporter 8, if you get antibodies against 2 or more of those, then the likelihood that you will get diabetes is very, very high. And it approaches 100%. It may take 20, 30 years, but it is very high. There will be some survivors, it's inevitable, and we like survivors, obviously, also to learn about why someone might be a survivor. These if you develop one antibody, always difficult to know what this means. Sometimes it's because they just developed the one and they didn't get any further, and that would be a nice thing to know. So if you could stop people at one antibody, that would be terrific. Or it could be that, you know, it's a chance finding in terms of these antibodies. The methods are particularly, they may be picking up something else occasionally, so not all of these may be the true antibodies that we associate with disease. The interesting thing, I don't know if it's nice, the interesting thing about progressing is that if you take these curves for the 2 and 3 antibodies here and put them into what we call incidence curves per year and look at how many of these multiple antibody positive children develop diabetes each year, it is reasonably consistent over time. And it's 11% in this cohort. So what does that mean? It means that if you're multiple antibody positive today, in the next year, you have an 11% chance to develop clinical diabetes. If you manage to get over that year and don't have it, the next year you start again and have another 11%, and then another 11%, etc. So every year, multiple antibody positive child will develop, has a 10 to 11% risk of developing clinical diabetes. Almost like it's a random thing. It's not quite random. We also know that, in fact, to get to clinical diabetes, often you go through a phase of impaired beta cell function. I showed it in that one example with various things that weren't quite right in metabolic tests. So that we also know that we can stratify. We can stratify that into, if you now have measured dysglycemia, well, it's somewhere up there. If you don't have dysglycemia, it's much less. So that's a useful thing to be able to do with people who are antibody positive, to try and stratify that risk. Important to follow them up. And there are also other things that probably change that we know. Age is very likely to be important. Older people who are antibody positive tend to go more slowly. So they're going to probably be down here. Girls go a little bit faster than boys. And we know that there are some genes that affect how fast you then go thereafter. Okay. I just wanted to give one example before I finish of how environment might be affecting the risk and the risk of autoantibodies. And this environmental factor is interesting because it is very, very strong protective environmental factor. Who in the family has type 1 diabetes is important with respect to the risk. And the important thing is that if someone has a mother with type 1 diabetes, their risk is around half of if they have a father with type 1 diabetes. Genetically, at least the DNA code is very similar between them. So it's unlikely to be straight genetics. It's got to be something else. And here you can see also that it's not just type 1 diabetes, it's the autoantibodies. So these are now development of these multiple autoantibodies. They have much less risk, especially in this early phase right there. That's where they're heavily protective. The protection is really specific for type 1 diabetes because in the same cohort, if we look at the celiac antibodies or the thyroid, the maternal type 1 diabetes does not protect at all. So it's very, very specific for the type 1 diabetes and the early phase of that. And what we know is that it's protective against development of those insulin autoantibodies. So they have a much reduced risk in this first year of life to develop the insulin autoantibodies. And wouldn't it be nice if we were able to find out how this works and so we could potentially not induce type 1 diabetes in a non-diabetic mother, but, you know, we could think of how that might be able to be used for protection. So they have very many things that are different to, so a mother with type 1 diabetes, a child who is born with a mother with type 1 diabetes has a number of things that are different to a non-diabetic mother. Often they're larger, etc., and they have, at birth and for a little time afterwards, high levels of insulin, proinsulin, etc. If you remember, insulin, proinsulin are key autoantigens in the disease, and they're the first ones that are targeted, at least from an antibody viewpoint. And what Jan Knoop has worked hard for in his, as a graduate student, he's just finished recently, is to try and understand what might be happening at the level of cells coming out of the thymus in these children who have a mother with type 1 diabetes. So he spent several years collecting cord blood, also placenta, but cord blood, and looking at the T cells that are present in these cord bloods, especially the ones that respond to insulin or pro-insulin. And so what he found was that, in fact, as might be predicted, if the children have so the offspring have a father with type 1 diabetes, he found quite a lot of these cells in the cord blood, so pro-insulin reactive or responsive T cells. But when he looked at children who had a mother with type 1 diabetes, they were much less than in that particular case. So what we'd like to think is that perhaps one of the mechanisms of this protection really is coming from a exposure to this insulin or pro-insulin. We don't know where, and that this extra insulin, pro-insulin, is perhaps a good thing at the level of the immune system that might be correcting some of the genetic susceptibility in these offspring. So to come to my end of my talk, just to summarize, there's a high risk of islet autoantibodies in the first years of life. There's a high risk of celiac disease autoantibodies in childhood, more in girls than in boys. Thyroid autoimmunity as teenagers and adults, again, much more in females. Children who are genetic risk or relatively protected against insulin autoimmunity if the mother has type 1 diabetes. They will develop diabetes if they have multiple autoantibodies, and of course they will develop diabetes faster if they are positive and also have dysglycemia. The importance of team, as I said, Annette and I work as a team. It works very well. Teams can be a great way to achieve things, and the collaboration between her group and our group has been a very wonderful experience. I also thank the Teddy, which is also another team who've worked hard to produce and hopefully understand some of what might be causing type 1 diabetes, and our many people who help us on the way with some funding. Thank you very much for listening.